Welcome back as we wrap up Chapter 11. So in this chapter, we've already discussed kind of the basic signal transduction pathway, but we've discussed kind of how it can end and kind of reasons why we do it. So right now we want to pick up and wrap up apoptosis, which is essentially going to be programmed cell death. We'll talk about that in the next slide, but we'll also talk about other uses for these cell signals, specifically sexual reproduction. When you look at a lot of organisms, we are going to send out chemical signals, and this works even with humans and other things that are they're mammals, reptiles, insects, where they can send out pheromones, which will be chemical signals that can then travel through, in some cases, pretty large distances. You're talking feet, or in some severe cases, you can talk much further, where you're talking hundreds of feet, perhaps even miles, where these pheromones can spread, and they can essentially send the message of oftentimes like the female being in heat, or some cases the males will mark stuff which can send signals when you're talking about mating to ward off other males or attract females. But there's lots of these signals that go out regardless when you're talking about sexual reproduction. Now for some organisms like yeast, who will be our poster child, because they're unicellular, they have an inability to use other senses to really figure out who they should have sex with. Because sex for them is going to be taking because they're haploid, so they typically only have one half the normal amount that we would have, or they have one set. They don't like we. If you look at our genetics, we're considered 2N because we have two of every chromosome. We have two sets. So chromosome 1, a specific chromosome, we have two copies, one from mom, one from dad. Well, these guys are only going to be N. They only have one set. So if they want to go through sexual reproduction, what they're going to actually do is just merge. And so they're going to be one larger 2N cells from merging, and then they can actually go do the process called meiosis that hopefully you guys remember, which is where it breaks apart to form a bunch of N cells from it, which can then give rise to their own unique separate organism. So for yeast, this process is going to be figuring out who you should merge with to create this new 2N, supercell, which we typically call a zygote. This is what happens after fertilization in humans, where we get the zygote. So they need to figure out who they want to form this with, and then afterwards they'll do meiosis to get back to their normal haploid 1N state, where they'll grow and live and be happy. And so for them, they don't have vision, they can't hear, so a lot of these senses are taken off. So what they're going to do is send out signals that kind of say what their gender is. And so other yeast can pick up on these signals, and they can figure out if the signal is opposite them. So if you're an A yeast, which is essentially a gender, you can think of it loosely like being male, if you will. And if you're alpha, that's the other gender, which if you want to, you can kind of think of as being female. There's no real sex organs. There's not really a male or female in general with them. But you can think of it as somewhat analogous. So if you're an A yeast, you would be looking for someone who happens to be an alpha yeast. And if you find that individual and you pick up the chemical signals that let you know that they're of the other type, then you can go through this process where you do sexual reproduction. But they can't do that without these signals because they don't know if they're compatible. So for them, signals are the only way that they can do things. They're not like insects, which send out pheromones, but we also have eyes. Animals in general tend to have eyes. They tend to have additional ways of kind of verifying what you are. And we tend to have pretty large differences where you can look at something and say, is that male or female? Not all things. Some birds, turtles, you can't. But for a lot of things, we can look at them and just right away kind of tell, is this male or female? Yeast can't do that. They don't have a lot of these differences in structures. It's more just a genetic difference. It's more at just a molecular level. It's not at like a larger, more tangible macro level where you can look at the yeast and go, oh yeah, that's a male. No. And so the signals are going to be how they get around this issue of not being able to otherwise detect the type of yeast that you are, so they can figure out whether or not it's okay, if it's compatible to go through this process of sexual reproduction. And then we've got apoptosis. I like to think of this as a Canadian way of doing things, where you politely, if instructed to kill yourself, do so in a way that's the easiest possible thing for your cell to then digest you and get rid of it. So it's kind of the polite way of going through and getting rid of yourself if you're a cell that's no longer needed or suspected of being in some way compromised, whether it's a virus, cancer. And so you would send this signal, and there are chemical signals that our immune cells especially can deliver to these cells that say, all right, You need to kind of hit the kill switch. You need to hit self-destruct. And so what will happen then is you have a series of enzymes called caspases that will go through, and they're going to start making the cell start to dismantle its DNA. So it's going to cut up its DNA to make sure that's not a problem. And it's going to start to form these blebs, which are just awesome names. Let's just all admit that. And these blebs will kind of start to shoot out, kind of like pseudopodia, if you've seen that in amoeba. And so they'll start to pull organelles in and just kind of start to form these extensions. And those extensions will then break apart into vesicles. Those vesicles now contain all the enzymes, anything dangerous that was in there, and then they can be cleaned up by our cleanup crew, which is typically our white blood cells or our leukocytes. So they can come through and digest these things to make sure anything that was in there is now gone. And because the cell's broken down its DNA, it's dismantled all of its machinery, anything that it was going to do that might have been dangerous should be stopped by the fact that we just pretty much destroyed all the innards, if you will, of that cell in the first place while it was destroying itself. So it's just a safe, convenient way of getting rid of things that might be dangerous, so long as they still respond to the chemical signals that say, time for you to self-destruct, please kill yourself in a very organized fashion, so we can then dismantle the rest of you and hopefully extract a bit of energy in the process. And then lastly, just our quick review then. So as we talk about things, I want you guys to remember that the key parts for a lot of this cell signaling, one of the first key parts at least, is do you have the receptor? I want you guys to realize that not all cells respond to all signals because many of them don't need to. When you're going through the process of puberty, you don't need to have the development of all parts of your body. You don't need to have all of your body grow when you just become huge. You'll have some growth, but in general you're trying to activate specifically the gonads to produce sperm, and you're trying to develop some secondary sex characteristics like facial hair that can advertise to females that you've reached sexual maturity and you can breed. You don't need to just go overboard with this. You know, females don't need to go overboard with this if you release estrogen. It's not like you need all of your body to just start swelling. You may have where breast tissue does because, once again, there's this advertising, if you will, that's going on. But it's not like your whole body should just swell and start loading up with tons of fat. We don't see that because only certain cells within your body have the receptors, so only certain cells are going to develop hair, are going to undergo chemical changes to allow for ovulation or the production of sperm. Only certain cells are going to go through this process where ultimately they will start to either grow or put on body fat or whatever, which allows us to get our end results of primary and secondary sex characteristics that make you male or female. The same thing happens with stuff like digestion, where you might say, all right, I want you to secrete this hormone, but you're telling it to the stomach because you want the stomach to secrete pepsin, which will help break down proteins. You don't want everything in your body just to start pouring out enzymes. So we can be very particular about which cell gets the signal just based upon who has the receptor. So it's kind of okay to dump a chemical like a hormone into the blood system that reaches pretty much every cell because most cells will likely just ignore it. It's only the cells that can use it that'll actually go through the process of signal transduction. And then we said this transduction process is where the cell gets brought in by this receptor and then it allows something else to happen, whether it's a secondary messenger, that's one of the things that can be activated, or we can go through and we can activate directly or secondarily protein kinases, which will start in many cases a phosphorylation cascade, where we'll have a whole series of things start to get activated by phosphorylation. And the whole benefit, we said, of doing this whole secondary messenger protein kinase pathway is it allows us to mess with the signal. So we can do things like amplify the signal that we couldn't do if it just went straight from receptor to the response. You know, it's a very direct process. We can't involve more molecules, whereas with transduction, we can take that one molecule and make it into many molecules, which allows us to ultimately produce lots of end molecules here. So in this case, it'd be like G, that will interact with the DNA. So we make sure that we really activate that gene, that we really transcribe it, or we make sure that we turn on all the enzymes that we have, and not just one of those enzymes and hope for the best. And so that's the importance of this transduction part. And then lastly, just the response, which keep in mind that's what we're actually going for. And it's most often going to be activating or deactivating an enzyme, or activating or deactivating a transcription of a gene. So essentially turning a gene on or off, which then affects whether we produce or don't produce a protein. which that protein may be an enzyme, so it can still be a similar effect where an enzyme's working or not because we're making it or not. But either way, this allows for us to then get something that changes within our body. It allows for us to do something that we previously couldn't because a new protein's activated, or it prevents us from doing something that we've been doing that's no longer needed. That ultimately should be cell communication in a nutshell. If you guys have questions, make sure you guys ask me tomorrow when we're in class. Enjoy your evening.